Okay. Assalamualaikum and good evening everyone. Sorry. Okay, so we just start the class today. Smaranda, have you signed in for your attendance?
Yes.
Can I proceed?
Yes, doctor.
Yes.
All right. Okay, so today we'll look at protein metabolism. So this is the overview for the lecture for today. So we briefly go through the importance of protein and the significance of protein, and then we look into protein metabolism. And we have actually covered this part, ketogenic and glucogenic amino acid, which is also part of protein metabolism. So for today, we will look into deamination, transamination, and also urea cycle. Okay. Okay, so what are the importance of protein? So mainly proteins are essential for cell integrity and also function. And the dietary proteins, meaning proteins that we acquire from our diets, are the primary source of nitrogen, which are then metabolized by our body. And these dietary proteins serve three main functions. So first of it, their constituent amino acids are used for the synthesis of body's proteins. And the carbon skeleton from the amino acids can be oxidized to yield energy. We will look into how these amino acids can eventually be an intermediate for Krebs cycle or TCA cycle, which are important for the production of ATP, energy. And then the carbon and also nitrogen atoms may also be used in to synthesize other nitrogen containing cellular constituents. Okay, so briefly the process of how proteins are digested and eventually metabolized. So protein enters the stomach, followed by stimulation of this hormone gastrin. So what will gastrin do? Gastrin will stimulate the release of gastric juice. So gastric juice encompasses of hydrochloric acid, pH 1.5 to 2.5, then pepsinogen, and also renin in infants or babies. Okay, so what are the importance of gastric juice components? Basically, hydrochloric acid to denature the protein and provide the acidic environment for the pepsin. And then these pepsin, which are secreted as pepsinogen, are activated by the hydrogen ion within the hydrochloric acid. So this activated pepsin will then cleave the peptide bonds of aromatic amino acid and also acidic amino acid. So for renin, so this renin will help infants to clot the milk. So this will convert the milk to a more solid form to prevent rapid passage of milk from the stomach. Okay. So when we talk about protein metabolism, one thing for us, important for us to understand is that there's no storage facility for amino acids in our body. So these amino acids are eventually incorporated into functional proteins, structural proteins, for example. And the amino acids in our blood and the extracellular fluid represent the amino acid pool. So we have like a pool of amino acid. And if the amino acids are not used to make functional proteins or structural proteins, they will be then removed from our body. Okay. So the metabolism of amino acid primarily occurs in the liver, but for some, for example, the branch chain amino acids, the metabolism occurs primarily in the skeletal muscle. And again, as I mentioned before this, the carbon skeletons of the amino acids can be used for gluconeogenesis, so the synthesis of glucose from non-carbohydrate sources or oxidized in the Krebs cycle. And these amino groups within the amino acid structure are also used to synthesis non-protein nitrogen compounds, which also used to dispose urea. Okay. So another thing that's important for us to understand of the concept of amino acid pool is that the protein content in the adult body remains constant. Constant in the sense that the nitrogen, there's a balance of nitrogen. The nitrogen ingested is equals to the nitrogen excreted or removed from our body. So protein synthesis is equals to protein degradation. So if let's say there's a condition showing that positive nitrogen balance. So it means that the protein synthesis is higher at a higher rate compared to degradation of the protein, and vice versa. Okay. Okay. So in healthy adults, the total amount of, this is distracting me. Okay. So in healthy adults, the total amount of protein remains constant. So constant means we said that the protein turnover, degradation, and also synthesis of protein occurs at equal rate. Okay. And this protein turnover is high in infancy, meaning in babies, because they need proteins for muscle build up, for example, for growth, and will decrease with age. Okay, so this is roughly, or the overview of the three process that will occur in protein metabolism. So we will look into one by one. So what I will do today is I will bring you through how this amino acid react with certain compound or chemicals, and also enzymes, and how the structure change, and how this protein metabolism relates to other processes in our body, for example, gluconeogenesis, and also Krebs cycle. Okay, so I'm not sure if you can see it clearly, but just to note that over here we have the skeletal muscle on the right, and this is the liver muscle. Okay, so for example, let's take an amino acid as the example to explain the protein metabolism. So for the sake of example, I'll take alanine. So this is the structure of alanine. So note that this is the carboxyl group, and this is the ammonia group. So in our skeletal muscle, there's abundance of amino acid. For example, here alanine. So what happens is that in the first step of protein metabolism, this alanine will react with alpha ketoglutarate. So alpha ketoglutarate is a keto acid that's also abundant within our skeletal muscle. So alanine will react with alpha ketoglutarate. So it's not just alanine. It's any type of amino acid will react with alpha ketoglutarate. Okay. So how does this reaction occur is that, so this enzyme aminotransferase will aid the combination of the reaction of alanine with alpha ketoglutarate. So what will this aminotransferase do is that it will cause the transfer of this amino group from the amino acid, for example here alanine, and this amino group will switch place with the oxygen group here in the alpha ketoglutarate. Okay, so now two new compounds are formed because only these two are exchanging places. So from alanine over here, so when this ammonia group is removed from alanine and the oxygen group is bind to alanine, so this will yield the structure called pyruvate. So where can we find this pyruvate? In which metabolism? Anyone? There's a hamster joining our class today. So where can we find pyruvate? In which metabolism? Fatty acid ke? Carbohydrate ke?
Carbohydrate.
Carbohydrate.
Okay, good. So please take note that, okay, now the reaction between amino acid alanine and also alpha ketoglutarate can yield one molecule of pyruvate. Okay, because there's a switch or there's a transfer of this oxygen group from alpha ketoglutarate to alanine and the removal of this ammonia group. Okay, so what will happen next to alpha ketoglutarate is that when oxygen is removed and this NH3 is bind to alpha ketoglutarate, it will yield amino acid glutamate. Okay. Okay. So this whole process is what we call as transamination. So transamination occurs when one molecule of amino acid react with alpha ketoglutarate, or we call it as a keto acid, and this process or this reaction is assisted or catalyzed by aminotransferase, So in this process, alanine and alpha ketoglutarate will exchange their exchange their group. So in this case, they will exchange NH3 and also oxygen group, and this will yield pyruvate and also glutamate. Okay, so that's transamination. Okay, so this whole process again is occur or takes place in the skeletal muscle. Okay, I have the written form of it later in this slide, so don't worry. Just go through with me so that I can make you understand the process. Okay, so this pyruvate, just a recap for everyone. So this pyruvate can eventually go into the acetyl-CoA or converted into acetyl-CoA, and this acetyl-CoA can go into Krebs cycle, which the main or the significance of Krebs cycle is to yield a lot of ATP. So this pyruvate is important for ATP generation. And also this for example, let me ask you, when will pyruvate turn into lactic acid? In which condition?
Anaerobic condition.
Yes, anaerobic condition, which occur when anaerobic condition takes place.
Rigorous exercise.
Yes, during vigorous exercise. So this pyruvate can turn into lactic acid. Okay, so what will happen to this lactic acid is that, so this is the written form of it. Okay, so this lactic acid can travel from skeletal muscle into the liver. So within our liver cells, we have this specialized enzyme called glucose 6-phosphatase. So if you recall the carbohydrate metabolism, so this lactic acid will eventually turn into pyruvate and glucose 6-phosphate. And because of this enzyme that is present in the liver, glucose 6-phosphatase, so this glucose 6-phosphate can turn into glucose. Okay, can anyone tell me what is this process called from lactic acid to glucose? What is this called? From the carbohydrate metabolism. Is it glycolysis or is it gluconeogenesis? Which one?
Gluconeogenesis.
Yes, so it's gluconeogenesis because it's the formation of synthesis of glucose from non-carbohydrate sources, okay, from the lactic acid that comes from pyruvate that comes from alanine. So this is gluconeogenesis, okay. So yeah, just take note that how this integration of metabolism occur, okay. Okay. So what happens to this glutamate over here from this alpha ketoglutarate? So this glutamate will travel from the skeletal muscle into our liver cells, and then this enzyme glutamate dehydrogenase will assist the conversion of glutamate back into alpha ketoglutarate. So because the enzyme's name is dehydrogenase, so it will add the water molecule to glutamate, and this is also assisted by NADP+. So NADP+ will be converted into NADH, and this glutamate, this enzyme will cause the removal of this ammonium group over here and switch it with an oxygen group. So now glutamate is changed back into alpha ketoglutarate. So where will this ammonium group go? So it will now be ionized and turn into ammonium. Okay. So this ammonium is toxic to our body. So we will discuss this later, how our body discard or remove ammonium. Okay. Okay. So this process of glutamate turning or converted into alpha ketoglutarate is called oxidative deamination. So the main enzyme here is glutamate dehydrogenase because it involves the addition of water molecule. Okay, so far can follow?
Yes.
Okay, so this is the written form, so you can go through later. Okay. So now we will focus into this reaction of ammonium that is formed. So this ammonium will turn into urea within the mitochondria in our liver cells or the hepatocytes. Okay. So now let's take another example. For example here, now that our liver from glutamate converted into alpha ketoglutarate, so this alpha ketoglutarate will proceed its reaction, or it will continue to react with other types of amino acid. So for example here is, I take this aspartate. So when alpha ketoglutarate react with aspartate through the same enzyme amino transferase just now, so there will be an exchange of this ammonium group and also this oxygen group over here. So alpha ketoglutarate will exchange with aspartate and it will form it will form glutamate and also oxaloacetate. Okay, so from alpha ketoglutarate, whenever it go through or binds to the amino transferase and react with an amino acid, it will turn or be converted into glutamate, and the another amino acid will turn into an intermediate. For example here, aspartate will turn into oxaloacetate. So if you remember, oxaloacetate is one of the intermediates within the Krebs cycle. So again, as you can see here, this protein metabolism can also enhance the Krebs cycle reaction, which in turn cause the high production of ATP. Okay, so this is just another example.
Miss,
Yeah.
can you repeat the one that enhanced the Krebs cycle?
Okay. So now that if you can see, aspartate is being converted into oxaloacetate because it has reacted with alpha ketoglutarate. So this oxaloacetate is actually one of the intermediate of Krebs cycle. So remember the Krebs cycle like this. So there's a lot of intermediates within the Krebs cycle. So oxaloacetate is one of the intermediate. So meaning that when there's metabolism of aspartate increase, oxaloacetate production will increase. So when this oxaloacetate increase, so this will enhance the Krebs cycle reaction. So when Krebs cycle reaction increase, there's an increase of ATP production. Okay, so there's increased yield of energy. Okay, is that clear?
Yeah, okay, thank you.
Okay. All right. Okay, so this one is just what I've mentioned before. So the amino acid metabolism is also important in the production of ATP, in a sense that the metabolism of amino acid can yield the intermediates for citric acid cycle or Krebs cycle, and also they can also yield precursor or intermediates for gluconeogenesis. For example, as we observed just now, is pyruvate and also lactic acid. Okay, so for example over here, okay, so the oxaloacetate from aspartate just now, metabolism of aspartate can go into the Krebs cycle and eventually produce ATP. So most of these intermediates are coming from amino acid metabolism. For example here, this is familiar, alpha ketoglutarate. And then this malate, one of the intermediate in the Krebs cycle, can also turn into or being converted into oxaloacetate, which can be converted into phosphoenol pyruvate, and this phosphoenol pyruvate can be reversed back to glucose. Okay, and causes so this process is gluconeogenesis. So this just to give the idea that how amino acid metabolism actually integrates with the carbohydrate metabolism and eventually helps in the increase of production of ATP, or help in the production of energy in our body. Okay. Okay, so now let's look at this toxic compound, the ammonium. So ammonium ion or ammonia is very toxic to our body. So we don't want this compound to be inside our body. So what will happen is that we will observe later on how this ammonium ion will combine with the alpha ketoglutarate and form glutamate, which is not toxic, non-toxic to our body. And then this ammonium can combine with glutamate to form glutamine via this enzyme glutamine synthetase. And this ammonium can also go through the urea cycle to form urea and eventually be secreted in our urine. Okay. So ammonia intoxication refers to the build-up of ammonium ion in the blood and also tissues. And the symptoms include impaired function of the brain. So when this ammonium are being transferred or transported into our brain, it will be converted into other compounds which can actually draw water into our brain cells. So what will happen? It will cause swelling of our brain, okay, and then cause tremor because there's a destruction to the nerve cells, ataxia, convulsions, lethargy, nausea and vomiting, and also slurred speech and blurred vision. And in severe cases, if there's high ammonium in our body, it can cause coma and also death because it's affecting our brain. Okay, let's take a 5 minutes break, can we? And then go back into the same link because the time is finishing. Okay, then we continue with
Okay.
video
Alright.
cycle.
Alright,
Okay, see you
Doctor.
in 5 minutes. All right.
Okay, Doctor.